Sexually dimorphic behavior, neuronal activity, and gene expression in CHD8 mutant mice. Autism spectrum disorder, or ASD, is a developmental disability that affects approximately 0.6% of the population, or 1 in 59 children. It is characterized by a variety of social deficits, including a lack of understanding or difficult communicating with their peers. Individuals with ASD may also exhibit compulsive behaviors or obsessive interests. One of the more curious aspects of autism is how it is divided amongst males and females. Males are 4 times more likely to be on the spectrum than females. This topic is of high interest to scientists who aim to understand this imbalance and vulnerability to ASD. When examining concepts that could account for the sexually dimorphic behavior of autism, scientists tend to start at the molecular level. CHD8 is a chromatin remodeling protein that regulates gene transcription. Its mutation is heavily associated with autism, accounting for 0.5% of all the ASD cases. While this percentage may appear low, it is important to note that there are currently over 1,000 ASD risk genes, so CHD8 makes up a significant portion of these. 84% of the individuals with ASD and the CHD8 mutation are male. Because of the vast sex differences associated with this protein, it was chosen to study in this experiment. Furthermore, the mutation of CHD8 produces the social behaviors of autism in mice, making it easy to study behaviorally and on a genetic level. In order to determine the underlying differences in behavior and prevalence of ASD, a variety of behavioral and genetic tests were performed on the mice with the CHD disorder. First, a characterization of the CHD8 mutated mice was performed to check CHD8 levels as well as overall brain morphology. Next, the social traits of the rats were tested with maternal homing tests as well as grooming observations. In order to further understand the behavioral abnormalities observed in the social test, CFOS signals were analyzed in both male and female mutated mice. In order to further view the genetic disparities between male and female mice, the hippocampal neuronal changes and synaptic connections were analyzed with the CHD8 mutation. To test these factors, currents were measured and immunoblot analyses were performed. Gene differences were further examined using RNA sequence analysis at P0 to test for transcriptomes in male and female mice. These genetic differences were then tested through gene set enrichment analysis, or GSEA. Finally, GSEA was used again with genes that are up or down regulated in ASD. Overall, the methodology in this study allowed the researchers to gain knowledge about both the behavior and genetic differences in CHD8 mutated ASD and how these differences translate in both male and female mice. Mice were separated from their mothers for some time, and during this period of time, observations were recorded. When the male CHD8 mutated mice were separated from their mothers, they were more vocal than the wild-type mice, as well as the female mutated mice. Furthermore, when the mothers were reunited with the pups, the male CHD8 mutated pups spent a greater amount of time with their mother, compared to both the wild-type mice and female mutated mice. When the mice were adults, they were tested for grooming during periods of isolation, with male affected mice showing greater amounts of self-grooming. These results demonstrate how a heterozygous CHD8 mutation leads to abnormal social behavior in male, but not female, mice. The figure shown displays the autistic behaviors observed in the mice. Mice were separated from their mothers for some time, and during this period of time, observations were recorded. When the male CHD8 mutated mice were separated from their mothers, they were more vocal than the wild-type mice, as well as the female mutated mice. Furthermore, when the mothers were reunited with the pups, the male CHD8 mutated pups spent a greater amount of time with their mother, compared to both the wild-type mice and female mutated mice. When the mice were adults, they were tested for grooming during periods of isolation, with male affected mice showing greater amounts of self-grooming. These results demonstrate how a heterozygous CHD8 mutation leads to abnormal social behavior in male, but not female, mice. The figure shown displays the autistic behaviors observed in the mice. CFOS is a marker of neuronal activity. Its levels were examined in male and female CHD8 mutant mice during separation from their mothers. When unseparated, male mice showed normal levels of CFOS, while females showed decreased levels. When separated, male mice showed increased CFOS levels in some brain regions and decreased CFOS in others. Female mice showed normal levels of CFOS. From this data, it was concluded that the responses by the mice were based on their baseline levels of neuronal activity. When testing synaptic transmission in the hippocampus, It was found that while excitatory transmission was normal, inhibitory transmission showed some interesting changes. In the CHD8 mutated mice, females had an increased firing frequency in CA1 neurons, whereas the firing frequency and amplitude decreased in males. Levels of excitatory and inhibitory synaptic proteins were found to be normal in both male and female mutated mice. Neuronal firing in the hippocampus was then tested for abnormalities, and it was found that the same opposite effect occurred, whereas males had increased excitatory firing rates, while females had increased inhibitory firing rates. When gene expression was compared between males and females, there were no differently expressed genes, or DEGs, found at P0. At P25, there were 96 DEGs found in female-affected mice and 3 in males. When David analysis was performed on the 96 female genes, it was found that these DEGs were associated with extracellular vesicles, known to mediate intracellular communication. Female DEG analysis also showed strong extracellular matrix, which regulate neuronal and synaptic development. GSEA analysis was then used to compare male and female brains. It was found that enrichment patterns diverged at P25. This suggests that at P0, when the CHD8 mutation is introduced, it causes small changes in a large number of genes, which show later at P25. More GSEA analysis show that genes in male and female mutated mice have gene sets that are similar to those in human autism spectrum disorder. The CHD8 mutation is one of the major autistic spectrum disorder associated genes, and its sexually dimorphic behavior makes it worth studying. In this research, it was found that CHD8 does in fact cause behavioral differences between males and females associated with autistic behavior. The males in the study expressed anxious behavior, which is characteristic of the CHD8 mutated autistic behavior. The females, however, showed fairly normal behavior. The researchers explored the levels of CFOS in the brains of females and males and concluded that the difference in their social behavioral responses are based on their baseline levels of CFOS. Now, not only does CHD8 mutation cause behavioral differences, but also changes that occur on the genetic level. In the hippocampus, females had increased inhibitory postsynaptic currents in the CA1 area, whereas males had decreased frequency in this area. Neuronal firing was then tested in the hippocampus, and it was again found that females had increased inhibitory firing, and male mice had increased excitatory firing. This opposite effect in male and female affected mice helps to explain why they behave so differently, even with the same mutation. It was found that females had a much greater amount of differently expressed genes than males, which were shown to be related to extracellular vesicles, important regulators of intracellular communication. The genes were also strong in extracellular matrix molecules, which are important regulators of neural development and functioning. At each age they were measured, the females consistently had higher amounts of DEGs than males. These genes, including their important factors, may help explain why females behave differently given the mutation compared to males. The researchers discuss how this is evident of a possible female protective effect. Certain downregulations and upregulations in the brain may allow the females to compensate for the mutation of CHD8 gene. GSEA analysis gave more insight to the sexual dimorphism of CHD8 mutation in the brain. At P0, there are not many differences between the male and female brain and gene expression, but the changes increase as the mice grow older. This suggests that the mutation begins with a small effect on a large number of genes, which displays greatly as the mouse ages. The results found in this study are fairly consistent with humans with ASD correlated with the CHD8 mutation. The study shows how the mutation behaves in both males and females and how the sexual dimorphism comes about. In the future, more insight about the underlying mechanisms of autism can help improve treatments and the well-being of individuals on the autism spectrum. In this paper, the sexually dimorphic behavior of autism was explored through the CHD8 gene. However, I felt that the study did not examine the effect of the CHD8 gene on females and autistic behavior. The females in the study, while affected with the mutation, did not seem to have any autistic behaviors that were described by the researchers. In fact, the researchers described a female protective effect that shielded them from many of the symptoms of autism. I would wonder if the female rat symptoms would even be enough for them to be diagnosed with ASD. If not, it would be interesting to see if the CHD8 mutation for ASD could be better modeled for females, or it would be interesting to further explore the differences between CHD mutated behavior in autistic males and females.